Already? Okay. Hi all. Good afternoon, evening, night, or morning, wherever you are. Before I pass on to introduce this keynote session, please be aware that the session is being recorded and that you're welcome to post your questions on the Q&A feature. Thank you all for joining this session entitled From Animal Health and Welfare to Plant and Crop Science, Contributions from Statisticians and Modelers at BIOS. And I'm going to start with a brief intro to our organization. Then I will introduce Karen, who will give a brief talk, and I will continue with Graciela, who will finish this session. At BIOS, or Biomathematics and Statistics Scotland, we are experts in data analysis, statistics, mathematical modeling, and bioinformatics. We apply our skills to real-world problems, and we put a focus on data science being not just a discipline on its own, but a fundamental part of the scientific process. We work in close collaboration with brilliant scientists from very varied fields, and we produce genuine impact on the world around us. BIOS in numbers can be summarized with around 15 members of staff across collaborating institutes in 3 Scottish cities, Edinburgh, Dundee, and Aberdeen. We have 18 women amongst our staff, 5 of them in leadership positions, 4 female associates, and 11 female PhD students. You can find more information about our organization in the QR code below, but also wanted to share that we are currently looking for a statistician specializing in animal and plant health applications, and this will be a permanent full-time position, will be located in Edinburgh, and if you're interested, please submit your CVs to before the 3rd of November. There's also another position that's gonna come up soon, so please keep an eye on our website. I'm now gonna move on to introduce our first speaker, Karen Keegan, who is a final year PhD student based at the Murdoch Research Institute with supervisors Dr. Eleanor Watson, Nuno Silva, Dr. David Longton, Dr. Karen Scott, and Nick Church from BIOS. Her PhD focuses on nanopore sequencing to investigate zoonosis and antibiotic resistance at the wildlife. And her talk's title is Optimizing Avian Fecal Samples for Gut Microbiome research via nanopore sequencing. So without further ado, I will pass on to Kate. Thank you. On to Karen, sorry.
Hi, everyone. Let me just share my screen. Can you let me know if you can see that okay?
Yeah, it's great. Thank you, Karen.
Okay, perfect. Okay, so thank you so much for inviting me to speak here today. So I'm going to talk to you about a portion of my PhD, which is looking at optimizing avian fecal samples for gut microbiome research and disease surveillance by nanopore sequencing. So just a little bit about me. I'm based at the Morden Research Institute predominantly, which is just outside Edinburgh. But I'm co-supervised by a range of supervisors. One of those is Nick Church, based at BIOS. So this talk as well is going to be quite a lot of lab focus, but there are data science, of course, in terms of bioinformatics pipelines that I'll talk about. So hopefully it's interest to a wide range of people. So just to give you the full sort of title for my PhD, so I'm interested in evaluating nanopore sequencing for looking at zoonoses and antibiotic resistance at the wildlife-livestock interface. So I'm based at the Foodborne Pathogens Lab at Morden, and we're predominantly interested in bacterial zoonotic pathogens. So these are things like Campylobacter and Salmonella, which are the leading cause of gastroenteritis and diarrheal disease globally in humans. And of course, alongside these bacterial zoonotic pathogens, we are of course, concerned about the emergence of antimicrobial resistance, or AMR, as this is a major concern for public health as well. And so my PhD sort of sits in the One Health framework, which is essentially looking at kind of zooming out and looking at the wider picture in terms of how pathogens move in the environment and how they end up, between animals, the wider environment, and potentially into humans, causing disease in humans. So I'm going to talk to you about one of the sort of main studies that I'm looking at in my PhD. And so this is looking at grey lag geese and cattle in the Orkney Isles. So Orkney, which is an archipelago just north of Scotland, it's renowned for its high quality beef production, and there's quite a lot of cattle on these islands. But over the last 30 years or so, the number of resident and migratory geese feeding and congregating on these cattle pastures has increased dramatically. And this has led to a lot of fecal contamination from the geese onto these cattle pastures. And the reason why we're interested in this is because we know that geese species, so wildfowl, are known carriers of the bacterial zoonotic pathogens that we are interested in, so Campylobacter, Salmonella. We also know that they can harbor a wide range of AMR genes. And essentially what happens is they can shed these pathogens through their feces into the wider environment. So one of our first questions was looking at whether what the pathogen carriage rate is in these wild geese, in terms of specifically these zoonotic pathogens, but also is there a potential for disease transmission between the cattle and the geese? They're in very close proximity to each other, and there's a potential that the cattle could, and vice versa, actually, the cattle or the geese could pick up pathogens from each other from their feces. So my PhD is focusing on using sort of advanced genomic sequencing technology, specifically Oxford nanopore technology, and really evaluating this sequencing technology as to whether it can be used to improve our disease and AMR gene surveillance in these sorts of wildlife-livestock interactions. So if you're not familiar with nanopore sequencing, it's essentially a long-read DNA or RNA sequencing platform. I'm specifically interested in DNA sequencing, as of course we're looking at bacterial pathogens, but you can use it for a range of different things. Specifically, how we use nanopore sequencing is through shotgun metagenomics. So essentially our sample is our feces from our wild birds. And what we do is extract the DNA from this fecal material and essentially sequence the DNA that's present. So in theory, we should be getting a range of different things. So we can be getting DNA reads that are from the host, DNA reads from bacteria, from DNA viruses. So we're really getting a comprehensive picture of what's in these types of samples. But the problem with avian fecal samples is that they are quite tricky samples to sequence from. They tend to have quite a lot of plant material, particularly in the case with these geese samples. And so one of the things I looked at was validating a DNA extraction method that actually worked with these types of fecal material, fecal samples, and that could also be used in nanopore sequencing. So what I did was I looked at 7 different DNA extraction methods. So these really ranged in terms of their cell lysis methods. So this is essentially how the cells are broken up to release the DNA that might be present. So we've got physical, chemical, enzymatic, or a combination of these. I also looked at different DNA isolation protocols. So things like the spin column method, which is quite a common method used. If you are a lab-based scientist, you might be familiar with QIAGEN kits. These are typically spin column kits. We did also look at magnetic beads as an isolation protocol. More traditional methods like phenylchloroform and anion exchange was another method investigated. And the sample inputs varied as well based on the manufacturer's guidelines. So this is the amount of feces used for each extraction method. So this ranged from 25 milligrams up to 500 milligrams. And so in order to compare across the different extraction methods that I looked at, I wanted to have the same sample types used for each method. So I'm quite lucky at Morden. We actually have a farm that has some domesticated geese on the farm. So this experiment was conducted before we collected our field samples from Orkney. So we didn't have any other samples to work with. So we had the domesticated geese samples, which worked really well, actually. So we had essentially what I did was pick up random feces and put these into 6 bags, randomly put into 6 bags. The contents of these were homogenized in exactly the same way. And then they were essentially, each bag was then a standalone sample that was used. So this was to generate enough material that we could use for each extraction method. And we also ran for every sample and for every extraction method, each sample was run in triplicate. So we needed to have a lot of material to compare. And then what I wanted to do was look at the sequencing output that you can get from the DNA extracted using these different methods. So what I did was I randomly chose one sample, which happened to be sample number 3. And for every extraction method, this sample was run on a nanopore MinION device, which is just pictured here. I'm not sure if you can see my mouse. You probably can't, but it's this sequencing device just up in the corner of the screen here. And this is just a small DNA or RNA sequencing device. It's pocket size, it's really, really small. And we do all our sequencing in-house. So I prepared the library myself using a rapid barcoding kit. So this kit essentially allowed me to multiplex samples. So I could have every extraction method, the sample number 3 from that extraction method run on this particular sequencing run. So essentially, once the DNA was sequenced, we had all this raw data. If you work with nanopore sequencing, you'll know that you tend to get quite a lot of raw data. And it's quite difficult to be able to store and analyze this type of data locally on your own PC or a workstation. So that led me to use a HPCC, which I'm sure some of you are quite familiar with. So this is just a high performance computer cluster, which is essentially just a remote collection of computers or clusters, a cluster of computers. And so this allowed me to upload my raw data onto the cluster. And then I essentially wrote and executed bash scripts as part of my bioinformatics pipeline so that I can analyze this data. I'm specifically going to talk about the taxa identification of the DNA reads, but I have also been working on antimicrobial resistance, a separate pipeline for that. But as time allows, I'm just going to talk to you about the taxa ID pipeline as it stands so far. So yeah, as I said, once you have your data up onto the cluster, you can write your scripts that do different things. So I have a little layout of the sort of pipeline that I've been working with. So essentially, one of the first things I do actually remotely from the cluster is check the quality of the raw data. And I do this in OR by looking at what's called the summary sequencing text files that are generated automatically from a nanopore sequencing run. So this just gives you information about the quality of your reads. I then take the raw data and I base call it using Guppy. There are more up-to-date software such as Dorado, which is Nanopore's newest base calling release. So this pipeline does also work with base calling with Dorado as well. But typically I've been using Guppy and super accuracy base calling. For those not familiar with what base calling is, just to quickly summarize that, essentially assigning bases to your DNA nucleotides, so ATGC, so that you have your full DNA read with your full nucleotide sequence. Once the sequences were base called, they were then a FASTQ file is generated from this base calling, and for every barcode, then these FASTQ files, because there's usually quite a few, are concatenated together per barcode, and then converted into a FASTA file, and then this is what's used for the BLAST analysis. So the BLAST analysis is just aligning your DNA sequences against, I use the NCBI nucleotide database, which is a really comprehensive database. And it also had various filters, filtering criteria. So hits had to be greater than 90% identity. The E-value had to be below a certain threshold. So there's ways you can swap in and out your different preferences based on how stringent you want your BLAST results to be. And then once the BLAST results are generated, they're usually in a text file format. So then these text files are then handled back in OR for the generation of figures, which I'm going to show you in the next few slides. So if you remember, I was talking about how we use a shotgun metagenomic approach. So this is essentially trying to sequence everything in your DNA sample. So what I first wanted to do to look at the different sequencing outputs from the different DNA extraction methods was looking at the kind of global picture of what's in these fecal samples. So I wanted to do that by looking at DNA reads per superkingdom. And your superkingdoms are your archaea, which we have here in red in this figure, bacteria in the orange, eukaryotes in the yellow, unassigned reads in the light blue. So these are just reads that were not given any taxonomy assignment. And then the dark blue are viruses, specifically DNA viruses in this case. But again, RNA sequencing is possible with nanopores. So if you're more interested in RNA viruses, this is also something that can be achieved with nanopore sequencing. So just to try to sort of illustrate what's going on in this figure a bit more. So on the x-axis here, we've got the different 7 different DNA extraction methods that I was looking at. We have on the y-axis here the counts, which you'll know they do differ in scale because we've got different amounts of things. So for example, bacteria, we've much more bacteria than anything else in this particular fecal sample. What I've done here as well is separate the read lengths into different read length categories. So we have read lengths of less than 1,000 base pairs, between 1,000 and 5,000 base pairs, and then greater than 5,000 base pairs. And again, this is sort of just illustrating the differences compared to Illumina sequencing in terms of the read lengths that you can get from nanopore sequencing. So essentially what I found when I looked at all these different read length categories and superkingdoms was that one method in particular gave the most reads per superkingdom at all these different read length categories, which was the MAGATRACT high molecular weight DNA extraction method. And this was a magnetic bead isolation protocol. I then wanted to look specifically at the bacteriophyla in these samples because, again, we're mainly interested in bacterial zoonotic pathogens that may or may not be present in our samples. So honing in on what's actually there in terms of bacteriophyla was an important aspect of this as well. So again, just to explain the figure here, so we have the 7 extraction methods on the x-axis and the proportion of bacteriophyla present on the y-axis. The total number of reads here just illustrates the total number of reads across for the whole sequencing run for each extraction method. So what I found was that almost every extraction method and the DNA that was generated from these and sequenced captured what's considered the core avian gut microbiome. So this would be your Actinomycetota phyla in pink, the Bacillota in yellow, the Bacteroideota here in dark blue, and the red is Pseudomonadota. Again, though, the DNA extracted and sequenced from the MAGATRACT high molecular weight DNA kit had the highest number of reads associated with these specific bacteriophyla. And what's more, the DNA extracted from this method also captured a more diverse range of bacteriophyla than any of the other extraction methods. So what I found was really a lot of the DNA extractions, in fact, the majority of the DNA extraction methods performed quite well. And what I didn't have here was QC in terms of DNA concentration per extraction method. So initially they all had very similar amounts of DNA, albeit quite low amounts of DNA, but quite similar across the board. But really the differences came about through sequencing the samples and actually looking at the how the reads were being assigned. So again, just to further emphasize that the MAGATRACT high molecular weight kit really had noticeably higher sequencing yield and did have higher taxa abundances across all the superkingdoms and bacteriophyla. One caveat though to this is that this particular method yielded a little bit less DNA compared to the other methods. And it did have less DNA when it was loaded on the library. So conversely, it's quite interesting that a method that had less DNA loaded on the library actually resulted in more reads. We did notice that there was more shorter reads. So this may be something to consider and does need to be looked at a little bit more in detail, because potentially that's a bias with this particular experiment, that method just had less reads and actually, or less DNA and actually resulted in more reads. So that needs to be investigated a little bit more. But it was great to see that we could use a method that did work with these types of samples and did give us some really interesting sequencing results. So the next steps and things that I've been working over the last few months are extract and sequence a lot of our Orkney geese fecal samples that we've been collecting over the last few years. We already have bacterial culture results from these. So we know samples that are positive or negative for specifically for Campylobacter, because that's the main pathogen that we focus on in our lab. So we do have that data. So it's what I've been doing over the last few months is sequencing samples that are either positive or negative for Campylobacter based on culture results. So these are the sort of gold standard in terms of disease detection in samples. So it's really comparing whether the nanopore sequencing gives similar or completely different results to the culture results, which is, as I said, the gold standard for pathogen detection, bacterial pathogen detection. And what I also want to do is combine the taxa ID results with the AMOR results that I'm also getting through my other bioinformatics script, and really look at whether we can link up AMOR genes on specific bacterial reads. So merging those two sort of data sets together and comparing and seeing if we can get that sort of level of detail as to where the AMOR genes are sitting on in terms of the bacterial species they're potentially in. So I think that's me. I'll just, yeah, I mentioned that already about culture. So yeah, I'm happy to take any questions. I did have a last slide there, but it seems to have disappeared. So I'll stop sharing my screen potentially. And hopefully you can also hear me.
Thanks very much. Yes, we can. Thank you, Karen. There's a question for you from Simona from BIOS. She's asking, thanks, Karen. Apologies if I'm being naive, but my science memory is rusty, but I don't believe that for a second. You said you have less DNA and more rates. One thing might be spurious rates, of course, but have you checked what is the best DNA concentration to load on your system?
Yeah, so we were basing the DNA concentration. So the nanopore library preps all sort of recommend a certain amount of DNA per sample, DNA concentration per sample. So for that particular method, for that library prep, the actual amounts were 200 nanograms per sample. All of my extraction methods yielded DNA. 6 of them were around 100 nanograms. That was the total that I could get. And the MAGATRAC method had about 25 nanograms, so about 4 times less. So I couldn't yield any more because those samples were actually spent at that point. That was the most that I could get from those samples, because they had already been ethanol precipitated down to increase the concentration of DNA. So yeah, it's a bit of a difficult one to understand. I have been reading, there's not much about why you would maybe get more reads given that you've put less DNA in. It is a bit converse to what you would expect, but it could be something to do with pore occupancy, that samples that are maybe loaded, that have a lot of DNA, are kind of taking up pores and moving more slowly, whereas ones that, you know, a sample like the MAGATRAC that had less DNA is potentially being able to move through the pores at a more faster rate, proportional to the others, and then maybe that's why we're getting more reads. But it is something that is a bit puzzling. And yeah, as I say, I need to investigate a little bit more. But hopefully, if anyone has any suggestions, if anyone works with nanopore sequencing data, it would be really interesting to hear, because there's not much in the way of, yeah, DNA concentration and optimum amount and yields that you can get. It seems quite variable.
Perfect. Thanks so much. Fascinating results. I have a quite a general question for you, and it's if there's anyone in the audience that is looking for a PhD opportunity, I would just like to ask you to give a reason for anyone to join BIOS to do a PhD with us.
Yeah, I mean, so my background is very much lab-based, which you probably could tell from my presentation as well. So I really didn't have much bioinformatics experience before I started, and I've really been able to learn so much through Nick, who's my bio supervisor, has been absolutely brilliant at helping me getting familiar with working with a cluster, first of all, and then learning, you know, how to write scripts, how to execute scripts, to troubleshoot, improve my code. So if you're someone who is maybe somewhat familiar with bioinformatics, has done a little bit, but doesn't feel very confident in it, I would say that BIOS is a great place to actually learn those sorts of skills in more detail in a really like supportive environment. I've never felt like, you know, I'm ever sort of judged for not having that strong sort of bioinformatics skill background when I started. And it's been great to learn it and feel like I've been, you know, getting more confident with it over time. So yeah, I think it's a really supportive environment to learn and to, yeah, really grow in terms of your skill set with coding and that sort of thing. So yeah.
Fantastic. Thanks very much. It's great to hear, Karen.
No
Very
problem.
best of luck for the rest of your endeavors. Yeah. Great. Now we'll move on to our second speaker, who is Graciela Martinez, a socioeconomic statistician at our Aberdeen site, who did her master's degree in mathematics at the National Autonomous University of Mexico, and who during her career has worked as social researcher in a private company and has been recently working on providing statistical advice in social research with researchers at the James Hutton Institute. Graciela, over to you whenever you're ready, and her title will be Understanding Byproduct Material Flows in the UK Seafood Industry to Estimate the Potential for Adding Value. Thanks very much.
Hi, thank you. Thanks, Altea, for the introduction. I just let me share my screen. Can you see my screen?
Yes, you just need to put it on full. Yeah, perfect. Thank you.
Okay. Well, I'm very pleasant to be here, especially because it's International Day of Women. And today I'm going to talk about understanding byproduct material flows in the UK seafood industry. Estimate the potential for adding value. This work is still in progress, and this work is in collaboration with Nasli Cosaglio and Simone Piras. Both are based in the James Hutton Institute, Social Science. And well, I'm gonna give the outline of this talk. I'm gonna talk about the background or the methodology, the results, especially I focus in the UK case, then for special fish species, then for Scotland, and then what is the next step for this work. Okay, the first part, the aim of this project is to estimate the scale of the potential gains from reusing fish and seafood in UK and Scotland. If you don't know, most of the fish that is produced in UK is catch in Scotland. So there is a interest from the seafood industry to try to estimate these potential gains. What is that? Okay, the purpose of this is to give some reuse or revalues of the surplus in the fish industry. For example, one of the uses for the surplus sea fish product is to use the viscera to produce fish meal plants. Another is use the bones, probably sell as a product, but as well probably I think it is started to explore, for example, use bones to produce some supplementary foods, and also the whole fish can be used as biofuel or composting. So there is a economic interest into quantify the fish the surplus in the fish production. One of the challenges that I found, in most of the cases, we need to integrate different data sources. In this case, I use the data that is published by the sea fisheries, organizations. As well, I use the His Majesty Revenue and Customs products. In this case, I face two different sources of data. So in this case, we have a different way to name different products. So the first issue is try to harmonize the products' names. The other challenge is we need to make some assumption in the absence of data. Most of the published open data is public, but so it's only given in an aggregated way. So in this case, we need to break it down each quantity. So in this case, we don't have this detail, so we need to make some assumptions. So I draft this methodology in order to estimate the surplus in the fish production. So in this case, I use two basic data sources. One of them is the UK Sea Fisheries Annual Statistics. And in the UK's Sea Fisheries Annual Statistics, they publish the live weight and the landed weight. The live weight refers to the amount of fish that is caught in the sea, but then also reports the landed weight. For example, sometimes some vessels catch the fish, but then none of the fish return to the port. So, for example, need to head on or go for the species. So there are two different measures. Then we have the exports. The exports are given for the His Majesty Revenue and Customs data. And in this case, the exports are reported in the final product. For example, the sardines can be exported as tin or probably as fillet. So in this case, we only know that the final amount of the final products, but we cannot, for example, estimate. For example, to produce a tin of sardina or tuna, probably you need to discard some part of the fish. So in this case, those parts are not reported, so we need to estimate. So in this case, I use these conversion factors. So in this case, the exports can be seen as two parts, the net weight and the surplus. The net weight in this case will be the final product. So in this case, we need to estimate how many the amount of the fish that was discarded in order to produce a product. So in this case, we use these conversion factors. The conversion factors are used to convert net weights on imports and exports in fishery in aquaculture products into lightweight equivalents. So, for example, this conversion factor can allow us, for example, if you produce a kilogram of fillet, probably you need 3 kilograms of raw fish. So in this case, this gives us an idea. For example, the total of feed that you need to produce a final product. So in this case, these ellipses are colored by the status of the data. For example, the orange one are available data, and the blue ones are not available data. So in this case, we need to make some assumption on the blue ellipses. Of course, it could be very the national consult, it can be inferred very easily. Just we take the difference between the landed weight minus exports. So this is like from this diagram, we can see that the distribution of the total fish can be seen as this. So the light weight can be seen as the not landed proportion, and also the exports and imports. But the exports can be divided by the net weight and the surplus. And we can assume and we can proceed in the same way for the local consumption. So all the product that was no export then was processed for local consumption. So in this case, we have the historical production in UK by the historical production for UK for different types of production. For example, the light weight is always greater than the landed weight because I was telling you some parts of the fish are discarded before to land it. Also, we have the exports. Exports net is a known number. So in this case, using the conversion factors, we estimate the quantity of exports. I was telling you the exports nets, they only publish the final products. So we need to extrapolate using these conversion factors in order to estimate the total, the quantity of fish that we need to produce these exports. One thing is that salmon was excluded from the analysis because in this case, we only interested in the sea fisheries because I think it's a key industry here in Scotland. And also salmon, most of the production is produced under the aquaculture production. Oh. Yes, sorry. Yes, the land weight and the landed weight is reported for the seafood fish, for seafood. So the thing that I observed is that salmon, like if we disaggregate this by species, salmon, it's not quite good described because most of the salmon was produced using agriculture production. And the other assumption that we made is that the local consumption surplus was estimated using the average consumption factor for each fish species. So based on this chart, we disaggregate each component by fish species. We focus in these fish species because these fish species are the most produced fish here in Scotland. And for example, the local surplus was obtained by the difference between the local production and the local processing. The local processing was obtained based on the local production divided by the conversion factor. One of the things that I observed, for example, for coats and monkfish, the exports exceeds landing. This cannot be possible because we cannot export more than we produce. So in this case, the exports were truncated to the levels of landings. And probably this typical value can be explained again by the origin of the system that was used to produce coats and monkfish. Probably our feeling is that probably coats also is produced under the aquaculture system. So using these assumptions, we can distribute this charge. Sorry. So based on this chart and under the assumption that we made, we can distribute the variable, well, the fish production in four components: the local consume surplus, the local consume net, the export surplus, and the export nets. Especially in this case, we are focusing the ribbons that are given in blue, no, in purple and green. And I was telling you, the aim is try to estimate the surplus of fish, especially in Scotland, because almost of the 70% of the fish production is originate in Scotland. But when I look at the data and try to look at, for example, the exports, the origin of the exports, it seems that the processing is no. For example, the most important port here in Scotland is Peterhead. So we guess that most of the fish is processed in this port. But when you look at the trade data, it seems that most of the fish is exported via England. So in this case, it's because of the bureaucratic pathway. So in this case, in order to estimate for Scotland, we estimate based on the proportion of workers that I employ in the sea fisheries. So in this case, it's approximately the 40%. So given this number, we distribute the total amount of the fish. So this is the numbers of the surplus, the potential surplus per species. And this is given by thousands of tons yearly. This is well, there is a lot of work that needs to be done. One of them that it's in this case we only be able to estimate this quantity, but the next step is try to give some value to each of this quantity. And also, imports can also be processed upon arrival in the UK. So we need to include it in the study. So for example, if some whole fish enter to the UK, probably they enter fillets when enter to UK. So we need to as well quantify the potential surplus. And additionally, we need to explore in the composition of surplus. And this is important because the market value differs from that one of the processed goods. For example, here, as you can see, where I can show, yes. For example, if you can see the space that are between the exports and the landed, sorry. Yes, for example, there is a larger space between this green one and the blue ones. It means that to produce herrings, you don't need to discard most of the fish. So probably you don't have the economical value of this surplus is not too big if you compare with the pelagic, because in this case you discard more to produce a final to produce the final product. So in this case, we need to estimate the composition of the surplus and try to give them some value. And yes, so thanks for your attention, and suggestions are very welcome.
Thanks very much, Graciela. That was great. Great work and fantastic talk. I have some questions myself, actually. I was wondering, because I believe this was a European project, whether there were any other countries doing similar exercises and whether the same issues are applied in terms of data integration and having to take quite a few assumptions.
At the moment, I'm not aware if there are other country.
Oh, perfect. And also, did you have the opportunity and the time to contact experts in this type of data to to try and reduce the uncertainty and around some of the assumptions that you've made?
Yes, there is a potential meeting in the future because, yes, we need to focus, for example, how is the way that each fish species is processed. So he as well, they can give us, for example, the value for each of the part of the fish.
Mm-hmm.
Yes, yes, yeah.
So is this work that is gonna be planned as part of the project or perhaps future
No, it's part
editions
of the project. Yes.
Oh, fantastic. Oh, great. I will look forward to looking at that because it's certainly challenging and a very interesting problem, I guess, in terms of integration and statistical analysis. So fascinating. Let me see if there are any other further questions. Otherwise, I'm going to ask you the same question as Karen, and it's just give us one reason why anyone should join BIOS and work with us.
Oh, well, I'm really happy. I've been working at BIOS for 2 years, and I think it's very collaborative team. Yes, I think the environmental work is very healthy, and yeah, it's a good place to work.
Brilliant. And have you found any barriers, or have you found differences between working in Mexico as a woman in statistics versus your experience here in the UK?
Mm, Well, mostly, well, if from my background, I work from in my previous work, I work for a market research company, a private company. So uh, I think the process to analyze data is under pressure. Is always a time issue, and here I think they try to prioritize the time. For example, you expect to produce a good analysis, but with less time, I think it's not possible. So I think I found very helpful. I think it's a good I don't know, it's a good point for bios. Try to prioritize analysis as well. If you have any doubt, we always have the opportunity to talk with each other to share doubts, suggestions. So I think I think it's the highlight for me is the collaborations among the staff. So it's not yes.
Fantastic. Thanks so much. Thank you ever so much again, Graciela and Karen, for great talks and a great session. And unless anyone else have more questions, that will be the end of our session. Thank you so much for listening.